Hello, everyone. Welcome to today's webinar. I'm Ann Thayer, CNN Senior Correspondent, and I'll be moderating today's event. This webinar is entitled Single Particle ICP-MS Overview of Environmental, Industrial, and Biological Applications, and it's being sponsored by PerkinElmer. CNN works with sponsors to identify topics of interest and value to CNN's audience and consistent with CNN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. A few practical points first. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you're disconnected during the webcast, please log in again according to the instructions you received earlier. You're encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I'll be posing as many as time permits. Please note that CNN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNN Online after the live webcast. The presentation today is being sponsored by PerkinElmer, a leading company focused on improving the health and safety of people and the environment. It does so through the development and delivery of technologies, services, and solutions that are used in applications across environmental monitoring, water analysis, and food safety, to clinical diagnostics, drug discovery, and biomedical research. The company is committed to advancing science, innovating, and collaborating with its customers. During the presentation, we'll hear from Chadi Stephan. Chadi holds a PhD in analytical chemistry from the University of Montreal. He worked as a product manager for QSAR Risk Assessment Services before he joined PerkinElmer as an inorganic product specialist supporting the various elemental analysis platforms. He currently manages the development of various nanotechnology applications that focus on measurement techniques for nano-object characterization using various analytical platforms. He has published over 20 peer-reviewed papers and book chapters. I'll now hand things over to Chadi.
Thank you, Ann. Good morning, everyone. Good afternoon or good evening, wherever you're joining today. We have a busy schedule today. We're going to be talking, we have a heavy agenda for the webinar. I will be briefly going over the theory of single particle ICP-MS for those who are new to this technology. I'm not going to be spending a lot of time on it. There's lots of publications available for review to know more about the theory of this new emerging analytical technique. I'll be mainly focusing on the application. I'm going to be looking at the three main application market. We're going to talk a little bit about the industrial applications. I've selected the three of the newest topics, looking at analyzing consumer product for nanomaterials. I'm also going to be looking at impurities in semiconductor grade chemicals and touch a little bit of how can we use this technology to improve slurry analysis when it comes to the semicon market. Then I'm going to be moving to the environmental applications. On the environmental applications, for those who are familiar with the technology, there's a lot of publication out there, but I'm going to be talking a little bit about how we are taking this platform or this technology and applying it to day-to-day real environmental samples. I'm also going to be talking about how can we use it to better improve our environmental implication when it's related to nanomaterials, better understanding of the behavior of nanomaterials in environmental matrices. And also, I'm going to be opening up a new topic about speciations, and it's something that I have made aware of through a group of researchers looking at the lost species, and that lost species could have been in a particulate form, and this is the reason why it's not showing on your regular chromatogram. And then I'm going to transitioning to the biological applications. On that, I'm going to be talking a little bit on how can we look for nanoparticles or nanomaterials in biological fluid, and try to invite you to attend our single cell ICP-MS webinar that will be following in the next month. Moving forward, if I am to look a little bit at the theory of single particle ICP-MS. It's basically introducing individual particles in the plasma of an ICP-MS. Once the particles goes through the plasma, it's going to burst into what we call an ion plume, and that ion plume is going to move to the detector, generating an instant pulse. Some people will call it a discrete signal because it's a sudden signal happening from an amount of ions getting to the detector. The success of single particle is related to the ability to integrate at a fast pace, what we call them, also known as microseconds dwell time, some people call it, or some researchers will call it fast scanning quadrupole. It's all related to the ability of integrating data in the microseconds, because this is where the events are happening. They're happening in the microseconds, and a pulse event generated from the nanoparticles have a duration of 300 to 500 microseconds. So if you are aiming to see it or to integrate it properly, you would like to have an instrument that is capable of achieving that fast acquisition. Moving forward to look at what this instrument can provide us with information, it's going to allow us to differentiate between the ionic signal and the particulate signal. It's going to allow us to count the particles, size them as it relates to the amount of matter from each pulse. If we look at the graph showing on your screen, to just give you an idea about how small can we see when we are operating in single particle ICP-MS. A 50 nanometer gold nanoparticles is only 1.26 femtogram. If we are to integrate all those ions and capture them all. So this ability is we're gonna use that information, or we're gonna use this capability of single particle ICP-MS to open up looking for impurities in semiconductor grade samples. On that, we're also the technique itself allows us to look for low particle concentration number, and it is great for environmental studies and environmental implication studies, because it's one of the only techniques or technologies that allows us to look at PPT and PPV levels of contaminant at the particulate level. The analysis is in a minute, so it's very fast. It's great for screening. It allows you to screen your samples for nanoparticles, and because it's a fast analysis technique, it also allows us to accelerate our understanding about what's happening to the nanoparticles. Do they dissolve? Do they aggregate? And that's in a quasi-real time. If I am to show you how this technology works, on your screen you should be able to see in animated. It's a screen capturing that shows you how the pulses are generated. So basically, if you see every pulse, and now I'm zooming into that movie, you see every pulse is basically a particles. If you look at the pulses, they are from different sizes, because in that solution we have actually 3 different sizes. We have a 20 and a 50 and an 80 nanometer particles. And if you look at the graph displaying on your left bottom corner, you will see the distribution being populated for you in real time. The x-axis is the intensity of the peak intensity in counts, and the y-axis is the frequency, so the counting statistics of how many particles of that intensity of that size we just captured or just been measured on our detector. A summary of the information that you will receive. You will get from each analysis, you will get the mean size of that distribution. You will get also the particle concentration for that specific distribution you're looking at. And also, as I mentioned earlier in a few slides, the technique itself allows you to differentiate between the particle fraction and the ionic constituting your background. So we apply an algorithm that allows us to automatically separate that background, and we calculate for you how much that background is actually equivalent in dissolved concentration. So you get all that information in a simple analysis time. Now in the next 3 slides, I'm going to be talking a little bit on the limitation of the technique, or what should you expect when you're using single particle ICP-MS. In this table, you're going to be seeing, I know there isn't a few numbers, but mainly 2 columns I would like you to look at. I would like you to look at the particle number and the particle concentration. So the particle number is a theoretical calculation or theoretical solution that we prepare the solution at, and the particle concentration is when we analyze the solution. And the last column in this table is the particle recovery in percentage. And if you look at it, we can achieve a great recovery between how many particles we prepared our solution to how many particles we are obtaining if we go down to around 1,000 particles per mil. Now, once we get to the 100 particles per mil, we start looking at our particle recovery. It starts struggling. In that example of the 100 particle mil, I've used actually 2 times. I've ran the sample for 5 minutes, and I've also ran the sample for 7 minutes. And if you look at the data, the more you run your samples, the more you're going to use your statistics to build that particle recovery. So if you are in the range of only 100 particles per mil, I do suggest on you to run your samples for an extended period of time, maybe 5 to 10 minutes. That's going to give you a good recovery number when you are looking at your data. And if I go down to the last section, the 10 particles per mil, over there it's really a game of statistics. Sometimes you're going to get that particle that makes it to the plasma, and you're going to get fantastic recovery. Sometimes you're not going to get it. So if you are using the instrument or this technology, or you are planning on running single particle ICP-MS, I would suggest on you to be in the range of 500 particles per mil, and that's going to be your media where you're going to achieve a good recovery on your particle number. So if I am to take that amount of particles and convert them to what a ICP-MS user is used to, because we usually use a PPT level or a PPB level of concentration. I give the example on the top, so converting those solution of 10,000 particles, 1,000 and 100 and a 10 to a PPT levels, just to give you an idea about how that translates into a numbers that you understand. So for example, 100 particle per mil of a 60 nanometer particles is an equivalent to 0.17 ppt. So if I am to take and look a little bit on the table below, the table below actually comes from a real samples that we were analyzing for iron impurities. The iron has precipitated in that solution in what we think is iron hydroxide. I'm going to be discussing that later on in my deck. But just to give you an idea, if we have an average size of a 30 nanometer and we have about 1,751 particle, I'm taking the middle example of that, that's equivalent to about 0.059 ppt. So this is kind of an idea about how single particle ICP-MS can open up an extra dimension in looking at impurities in chemicals, only if they are in a particulate level or in a precipitate level. The other section, as single particle, it's a technique that counts the particles, which we went through. How many particles can we count, and how we are efficient in counting those particles. There's also how small of a size can we achieve with single particle ICP-MS. For that, it all depends on the operating conditions and depending on the user ability to tune the instrument for the analyte of choice. But one specific or a few specific things that I would like to stress: the density of the materials that you are using is important and is very important to understand, because the instrument, it's a mass technique, so it's going to measure the amount of mass from that pulse, and it's going to translate it to a size using the density. So the density is very important, and the other thing that you want to think about also is the mass fraction. Is that particle totally made from that analyte or not? Is it an oxide, or it's a pure metal? So all that it's important to understand. But I'm hoping that this slide kind of gives you an idea about what are the sizes that you should expect from a detection limit perspective. Now, moving forward, as we are using and constantly improving on single particle ICP-MS, single particle ICP-MS is taking a depth into standard methods On the slide, you will see an effort that has been led to develop a standard and an ISO standard method for sizing and counting inorganic nanoparticles in aqueous media using single particle ICP-MS. This standard method is in the work. It is almost at the final stages. We're hoping to have it very soon released. Similarly to the ISO, going back to the American standard, the ASTM, we will look at a few of the work items that are being used related especially to silver in textile and silver in consumer product, and all those methodology that will have a section where it can be used single particle ICP-MS towards investigation if that silver in that textile is in the particle format or not. And I will be giving an example in a few slides through a work done through one of our collaborators. So that was concluding the section that I'm going over the theory of single particle ICP-MS. I hope it is for further information. There's a lot of publication available on Elmer website or in many scientific journals. In the next section, I'm going to be looking at industrial applications. On environmental, or focusing on the application itself. From an application perspective, I put them into different silos. We have environmental, we have consumer product, we have biologicals. In the environmental, there are so many application nodes available on PerkinElmer that touches on the environmental angle or on the consumer product. Unfortunately, for the sake of time, I cannot cover them all in this webinar. I've only selected a few. But if your samples are related, for example, to a surface water or drinking water or bioavailability studies, please do consult PerkinElmer website. There is probably an application node that talks about single particle ICP-MS in that matrix that you're looking for. So in the industrial, I'm going to start with consumer products. The first example that I'm going to be talking about is nanoparticles in sunscreen or cosmetic product. The example talks on sunscreen, but there is other nanoparticles or particles used in many of the cosmetic. It is available in the application node, and also we have a published article on it. The highlight of this work really it is using single particle ICP-MS to quickly identify if that sunscreen has titanium dioxide in it, what are the sizes of, or what is the distribution of titanium dioxide in that sample, and also back calculate the titanium content from titanium dioxide particles to how much percentage of titanium that is in that sunscreen. Mainly, and the big improvement in analyzing cosmetic products or sunscreen for titanium dioxide using single particle ICP-MS is avoiding sample preparation, because sample preparation, when we wanted to get the total content of titanium, would have required a digestion, a complete digestion using acids, mainly sulfuric acid, or for that matrices. For that, analyzing it in single particle ICP-MS will allow you to simply prepare your solution through a surfactant, analyzing it directly on a single particle ICP-MS. The second example in the consumer product is analyzing for many of the nutraceutical or the multivitamins. In this application node, we looked at a few products available on the market that contain silver colloids or silver there is mesocolloid or mesosilver they call it. Again, it's a quick test from a quality control if you are a manufacturer or if you are a regulatory body, you want to test what are the particles in that product. Are there really nanoparticles or particles in that product? What is the dissolved fraction? It's a quick and easy test to do. It's a great technique to be able to look into the usability or the implementation of nanomaterials in many consumer products. The next consumer product I'm going to be talking about is clothing, silver in textiles, and how we are able to look at the efficiency of the product. For example, if I am to include silver particles in a textile and put that into a various washing cycles, what happened to the silver in that washing cycles? And in this slide, and I mean, if I can bring your attention briefly to the graphs, if you look on the example, there is a color detergent on the top and there is an oxy detergent on the bottom. We added silver we wash them with a silver nitrate, and you will look, you won't see much. But if you add particle addition to the color detergent, you see the particles are where they are. And if we are to add silver particles to the oxy detergent, you will see that the oxy detergent is basically dissolving the silver particles. So having a textile embedded with silver particles and washing it with an oxy detergent will probably wash the silver off that textile. The next example, and that's conclude the section for consumer product. Now I'm going to go and look a little bit at the industrial or impurities in semiconductor grade chemicals. One of the examples that I'm going to be looking at is iron. If we are to understand iron into some of the solvents, and I have on this slide a chart representing the solubility of iron versus the pH. If we are to look at iron at low concentration, at base pH, iron tends to precipitate in high iron hydroxide. So how does that translate to some of the chemicals used in the semiconductor grade? If they do have some iron, even at a very trace level, that iron will basically precipitate, and that iron will probably cause some issues for that industry. So very recently, we've put an application note on that. The application note look at running a few chemicals, few solvents, basically at the base solvents. And we are looking at particles or precipitate of iron, and we are actually using this technology. We're using single particle ICP-MS to be able to provide a better control, quality control on the purity of the chemicals. And as I showed earlier, if we are to only find a few particles in that semicon grade solvent, if we are to translate it to a pure concentration, it's basically in the sub-ppt levels. So if you might be running this solvent in a regular ICP-MS, and it's showing that it's below your your detection limits. But in reality, it is below your detection limits from the ionic fraction. However, it might contain some particles or some precipitate of iron that you're not able to see and identify. And this is where single particle ICP-MS comes into play, into investigating the impurities of some of the semiconductor grade chemicals. The next example, it's another solvent. So what we were just showing at tetramethyl ammonium hydroxide, the TEMA. In this example, I'm showing the PGME. The PGME also it's a base. It's going to look at, and it's going to give you and provide you with scanning for iron between, we wanted to see if it's coming from cyclohexanone or it's coming from PGME. So we mix PGME with DI water, the graph on the top, there is no spikes. Mixing it with cyclohexanone, you start seeing there is some iron spikes forming up in that mixture. So now we are concluding that the iron is coming from the cyclohexanone. And the cyclohexanone also, it's slightly on the basic when we are looking at the pH. I think it's at the pH 8 or 8.5. So building on this application, Looking at impurities in silicon grades, one of the recent projects that I worked on is trying to be able to look at silicon dioxide precipitate in sulfuric acid, in pure sulfuric acid. It's something that if you'd have asked me before, it's like, can we see silicon dioxide in sulfuric acid? It's like I would have thought, it's like it's an acid, you won't have any particles in it. But again, we all know that for us to be able to dissolve silicon dioxide or to achieve a proper digestion, we usually tend to use HF. So silicon dioxide, if we are to look at it in various acids other than HF, we might be able to find. And in this project, we were looking basically at the purities of silicon dioxide, and the purities of different batches of sulfuric acid, looking at silicon dioxide as a trace contaminant. And single particle ICP-MS were very, very good in that project. And if we were able to differentiate good batches from bad batches using silicon dioxide as an indicator. One of the last topic that I would like to talk about when we are looking at impurities in the semiconductor grade or into that semiconductor market, it's not really related to the impurities itself. It's mainly related to the slurries. Slurries are such as cerium oxide or aluminum oxide are mainly used in the polishing process of many of the semicon, of many of the wafers. For that, we would like to have as tight as we can control over the size and the size distribution of that slurry. So basically, we would not like to have any large particulate or large clusters in that slurry, and that's going to define how well and how this slurry is beneficial for our work. So single particle ICP-MS, because of its ability to investigate particle by particle, so basically all the information that single particle ICP-MS offer us is basically particle by particles, not like other light emitting technologies where the information is basically the average of that light received from that sample. So single particle ICP-MS, it's opening up a door into improving the quality control of slurries when we are analyzing them into a single particle mode. Because now, for example, if we have one large cluster and 1,000 particle, or that's a ratio, we are clearly able to be able to see that, or at least trying to investigate if there is large particles or not. And that's conclude with that information. I do conclude the section related to the industrial applications. So we covered some of the consumer product and some of the semicon impurities, or impurities in semicon grade solvents. There's other, and there's, as I mentioned earlier, there's many other applications available on PerkinElmer that touch the industrial section. We're moving towards the environmental applications, and the environmental applications I'm going to be focusing mainly on drinking water and drinking water treatment process. It's an ongoing work. We started about a year and a half ago, where we started looking about, how well our drinking water treatment centers are when it comes to removing nanoparticles. So the initial work was documented looking at silver and gold and some of the titanium dioxide. So we looked at the different stages. So for example, we spiked what we call them the source water. So basically the incoming water, whatever we're using, we spiked it with nanomaterials, and we were able to identify them, and we tried to follow the various steps. And we came to the conclusion that the flocculation do remove most of the nanoparticles, because it is actually the step, the flocculation or the coagulant removes most of the nanoparticles, as nanoparticles tend to behave very similar to colloids, and these are the steps designed to remove colloids. But then we wanted to take that more further, and we wanted to investigate into commonly used nanoparticles, commonly found nanoparticles in environmental matrices. So we started to look a little bit more into titanium dioxide and cerium oxide. Titanium dioxide mainly been used as a sunscreen, as we saw, and we tend to use a lot of sunscreen. It's one of the most used whitening agent as well. So we use a lot of titanium dioxide, and we wanted to investigate. So we took many source waters, and if you are to look at titanium or to look at titanium dioxide, I can have guarantees for you, you're definitely going to see some titanium dioxide in your river water or in your surface water. It's just there. Very similar to cerium oxide, but cerium oxide has a different source. It has a natural source from erosion of rock. So cerium to lanthanum, I will talk about it briefly in the next few slides. Could be a potential, and the challenge is going to be differentiated between naturally occurring cerium dioxide and anthropogenic source of cerium dioxide, and it's something we'll probably take a different discussion. But if you look at the outcome of the slide, that lime softening removed about 90% of the natural and engineered nanoparticles. So this is good news. And if we are to go to the coagulation step, and we increase the efficiency of removing the excess of nanoparticles. However, what I wanted to bring your attention to is that during our studies, we broadened our studies. As first, we looked at 6 different municipalities or drinking water facilities, and we started looking at their source water. So we came to a few things that I think it's a good thing to know, that we definitely can find titanium dioxide in river water and in source water. However, we couldn't find them very well in well water. So the well water, everything that is naturally filtered, they tend to remove, or the amount of spikes or nanoparticles in that well water were less. Cerium dioxide, or cerium dioxide, was found. We also tried to add lanthanum because the differentiation between cerium dioxide from a natural source will have to have a ratio to lanthanum, and if it is from an anthropogenic source, because it's synthesized, it's basically assumed to be into a pure cerium. So we start looking at that differentiation, and it's an ongoing work that we hopefully can talk about it, and we'll have some papers on it in the near future. To bring you back to digging a little bit further into the drinking water facilities, and mainly on what we call the zone 4, which is the coagulation process. In the coagulation process, and looking at different type of coagulants. So we have aluminum, we have some of the ferric, and we have a polymer-based coagulant. And if you look at the graph, you're looking at titanium, you're looking at cerium, you're looking at zinc. What I want to be able to bring your attention is to the graph on the left, mainly on when we are adding polymer to the aluminum coagulant, and you start seeing that there is a difference between titanium behavior to zinc behavior to cerium behavior. And this is kind of bring us to the question, and there is also a difference between the river water and the lake water in the removal process. So what I'm trying to get into this over this few slides is that particle removal in drinking water is dependent on the source water. It's also dependent on the treatment. And also, I wanted to be able to tell you that it is particle specific. So titanium dioxide will behave slightly different than cerium dioxide, and will be dependent on those angles. Shifting a little bit gears from the drinking water, I'm going to go and tell you a little bit about the recent work I've been working on using ICP-MS, or the single particle ICP-MS, to investigate the behavior of nanoparticles in culture media. So when I say culture media, I'm talking about moderately hard water, I'm talking about hard water, I'm talking all those culture media that environmental toxicologists use to study the effect of contaminant on microorganisms and so forth. Now, in the question or in the instance of investigating particles or nanoparticles, three things could happen. We want to know few of the many things. First of all, we want to be able to conduct our experiment at a particle concentration that is of an environmental relevant. So we want to be able to do it at the PPT and the PPB level. I want to be able to investigate if these particles are staying intact or they are going through the two phenomena, dissolving or agglomerating. And this is where single particle ICP-MS really shine, and it is one of the most prominent and promising techniques into that field. So let me move a little bit in the slides, and I will try to explain to you a few of the things. In this next slide, we actually used a core shell, a nanoparticle system. The core is gold and the silver is a shell, and we did that intentionally. We know that silver tends to transform in culture media, so we want to be able to provide a view into its transformation, but also we wanted to be able to use the gold, the core, as a traceable. So we wanted to be able to use the gold as things to track the agglomeration, but we wanted to use the silver to track the solution. So it worked out to be a very good system for that. The next slide has a lot of information. It compares light, dark, various concentration, various temperature. My intention, I'm not going to dig into details. I just want to tell you just briefly, if you look at, for example, at the light, the two graphs on the right, on the left-hand side, the top is 15 and the bottom is 22 degrees. And only by changing the temperature of 2 degrees, you look at silver, which is the first column, how it is, how they are different. You see that when you are at the 22 degrees, the solution of silver is basically accelerated. It's more prominent than when you are at the 15 degrees, and that's the line. And then you can go and take into different things. And if you look at the gold, and that's at the 100K, right? If you look at the gold, you see the core, it's stable. It's nothing. There's no changes in that, so there's no aggregation. Basically, it's only the solution, the difference into the light between 15 and 22 degrees. Now, I'm going to move to the next slide because it's going to be probably slightly more visual for you to be as an audience to be able to see. In that slide, you look at three different concentrations. The 44 million particle per mil was chosen because historically, and if you look at a lot of the existing papers, they are all done at levels or at concentration that are really very, about 1,000 times more than what you can see and find in the environment. And that's because they are limited to instruments that couldn't go that low. So they were using a light emitting instruments or so. Now you have single particle ICP-MS can bring you down to investigate your particle behavior at an environmental level. So you can go to the 10,000 particles, and if you are to compare the 40 million to the 10K, for example, and if we are to look just at the particle number, you will see that 40 million, the particle number is decreasing. When you look at silver and gold, this is a suggestion that agglomeration is happening, so the particle numbers is reducing. They are agglomerating. If you are to look at the 10,000K, you will see the gold, which is a green bar, it's flat, there is no aggregation happening. But if you look at the silver, you see the particle number actually is increasing. And silver tend to do that. It tends to dissolve and reprecipitate at the lower size. So if you are to operate at that low particles, you are completely having a different behavior of those nanoparticles in that culture media than when you were doing your studies at an elevated concentration. So it's very important to get that understanding about the importance of using single particle ICP-MS when you're doing environmental studies. The next slide basically summarizes what I just went through by comparing various concentration of nanoparticles looking at the dissolution and the aggregation. So if we see the bar going up, that means there is an aggregation happening. If there is the percentage going down, that means there's a dissolution happening. You will have a gain and loss. So aggregation is basically the predominant phenomena at elevated concentration. And while you look at dissolution followed by precipitation is the dominant phenomena at lower concentration. And this is mainly for silver that you're encountering that, because silver is really the outer, consisting the outer shell. It is the analyte interacting with that media. Moving forward, I'm going to switch a little bit topic, and I'm going to talk a little bit about the speciation, or having particles as your lost species when you're doing speciation work. And I came to that through a recent presentation that was done by the group from University of Aberdeen. Basically, they were looking into the reason why these pilot's whale end up on the shore, and they were investigating and looking at selenium and mercury's content in both the brain and the liver. And we're looking at, and they were doing some investigations and using some speciations, because that's basically what we do when you're looking for selenium and mercury. But there was always that unknown species. There was always that negative equation where the total amount of selenium is never equal my species. So the team over there investigated a little bit more. So they prepared the samples through an extraction, and then they went into analyzing it by single particle ICP-MS. And if you look at the data, you sure think you can start seeing some particles or some spikes, and those spikes correlate to a particle size. So you might be looking at doing speciation and doing toxicology in a way, but ideally that lost species, especially maybe in this case selenium and mercury, it could be in that particle format that you're looking for. So having single particle ICP-MS to be able to look for that lost species, it's a great beneficial. I mean, I think the topic is great, and it's very innovative and new. So further on their finding, and looking at the graph, you will see that adult whales have significantly more particles of mercury and selenium than juvenile whales. Maybe the absorption happened as regular mercury, that the mercury is transformed to a form of solid mercury and a solid selenium into that organ. And there was no difference between the different organs. So basically both mercury precipitate and selenium precipitate were found in both organs at equivalently. And on this topic, also we looked at the sizes. And because single particle ICP-MS tend to give you not just the particle number, but also the size. And if you look at the average size seems to grow with the age. So it's basically what it suggests, that this deposition is basically happening inside of the brain of the animal. So it might come inside as a regular ionic mercury, but it might be precipitating into those organs and grow over time. So you may be able to look a little bit about particles when you are doing speciations. Moving forward, I'm going to talk a little bit more on the biological applications. So that concluded my environmental section. Again, there's so many other applications related to environmental use of single particle ICP-MS available on PerkinElmer website. For the biological applications, I'm going to briefly glance over it. The first thing that we did is basically, can we see nanoparticles in biological fluid? We took some blood, we took some urine, and we spiked them with some nanoparticles. And yes, of course we can, because it's a liquid matrix. You can precisely count particles, and you can precisely size the particles. And the analysis is basically straightforward, very similar. You take your blood, you divide it 20-fold, and you analyze with your diluent, and you analyze it. And if you have particles in there, you'll be able to see them. And similarly for the urine. That was the first thing that we did. The second application node that we worked on it with in SPQ, our collaborator in Quebec, and we actually started looking at the long term, and we'd like to look what's happening to the nanoparticles into that biological fluids. So two things we've actually started doing it, started working on. The first thing is we looked at the long-term behavior of gold in blood and urine, and if you will see that gold is pretty stable in blood, and gold is pretty stable in urine. There's nothing happening to the gold, both from a size perspective. You might tend to see some loss into agglomeration, loss in the particle number, and this is something that we are currently investigating. But if you look at silver actually have completely different stories. In these two slides, you will see even at the short term, because the gold data were in days, now we are looking at the short-term transformation, silver actually tend to start dissolving in blood, and definitely at a lower rate than it will dissolve in urine. And if you look at urine, the dissolution is it's more predominant. It's about 3 times faster that is happening in blood. So if you are looking at a tracer and a marker for to look for nanoparticles in blood, in urine, single particle ICP-MS could be the instrument that you need to investigate, or you need to look at it, its capabilities. The next thing that I'm not going to be talking about it today, I'm going to be talking, we're going to have a dedicated webinar for that. It's going to be aired on March 22nd. 2016, of course. In that webinar, we're going to be looking at single-cell ICP-MS. We're going to be using this discrete capabilities of detecting particles and applying it to cells or cell culture or individual cells. Algae works, yeast cells, bacteria cells. Mainly, we're going to be focusing on cancer treatment and having our guest speaker, Dr. Lauren Emable from NIH. So I'm not going to be talking about it. We're going to have a dedicated webinar for single cell analysis. On that, I would like to take a minute to thank all those who contributed to the content of this webinar. This webinar could not be done by a single individual. This webinar is through many collaboration ongoing on a global level. From that list, the list is populated in front of you with Dr. John Olesik from Ohio State University, Denise Mitrano from the Switzerland Federal Laboratories, Dr. Rashid's group from Missouri State University, Dr. Lee's group from University of South Carolina, Feldman from the University of Aberdeen, Cyprien from NSPQ. And I'm sorry if I didn't use lots of doctor in front, because they're good collaborators. We work with them on a day-to-day basis. And of course, many of my colleagues at PerkinElmer, Dr. Lee Davidowski and Ken Neubauer, who've contributed a lot to the content of this webinar. My last before slide, I selected some of the papers. Some of the data that are in this webinar or that were discussed in this webinar are available in application notes. Some of them are available in peer-reviewed journals. Some of them are up here. And I really do encourage the audience to visit PerkinElmer website and look at application notes. In application notes, it's a very simple few-pager that discuss what are the instrument conditions and how you can sample prep your samples to achieve better results when you're analyzing your samples. With that, I would like to thank your attendance. If you have any questions, I'm sure that we get some question by the end of the sessions, but if you have any questions and you're looking for any information, you can find please, my email is up here. Feel free to drop me an email, and I'll be happy to answer your questions. With that, I will hand it back to Anne. Thank you.
Thank you, Chadi, for such a great presentation. We do have some time for some questions, and we've had a lot come in. One of the first questions goes back to the silicon dioxide. Is 1 molar HF concentration enough to dissolve silicon dioxide, and does it require any heating?
Well, I won't be able to tell you till I try it. What historically been digesting silicon dioxide is when we want to achieve a proper digestion, we use a microwave with a concentrated HF. This is the way that I used to follow. Again, to answer the heating process, yes, probably heating will help the digestion. However, is 1 molar enough for what amount of silicon dioxide? That's something that they need to investigate further.
Okay. Another question, is the ability of single particle ICP-MS due to sample intro software processing of the signal from the ELAN or Nexion, or is there a hardware software specific for single particle?
Well, if I understood the question correctly, I would say the sample intro is a standard sample intro. Now we can go and use a high efficiency sample introduction system to improve the number of particles that makes it to the plasma. We might want to think about is autosampler that stir the solution if we are doing single cell. So these are the things specific to the sample itself. However, the data processing or the data representation is basically related to the hardware speed, what we call them data acquisition, so the speed of data acquisitions. So it's really a software processing signal.
I have another silicon dioxide related question. For the application of silicon dioxide nanoparticles in concentrated sulfuric acid, did you see any size lower than 100 nanometers?
As we are working with it, I think the best that we are able to achieve during that day was a detection limit, if I recall, about a 90 nanometer. But when we're talking about a 90 nanometer detection and we are running an unknown sample, you want to be able to be into the 100 zone for you to be able to, or 100 or 110, for you to be able to start quantitating or being comfortable in that quantification process.
Okay. We have another question. Is iron measured in, they have written STD, which I'm guessing might be standard, or DRC mode, and which isotope was used?
Okay. The iron was used, okay, so it's a great question. Iron was measured in DRC mode, dynamic reaction mode, using ammonia, and we were using mass 56, the most abundant isotope for iron. The reason we used that is because we wanted the most sensitivity. So the reactive gas will handle reducing the background, and we are able actually to use iron at mass 56 to achieve such measurements. However, also I would like to mention that collision techniques does not work well, or collision approaches does not work well when we are looking for single particle ICP-MS, and that's mainly predominant by the collision approach because you lose a lot of energy, a loss of signal when you're doing collision analysis. So basically for single particle ICP-MS, the best route to go is to use a reactive gas.
Someone asks whether single particle ICP-MS requires sample digestion.
Well, it requires sample preparation, depending on what you are trying to achieve. Because the particles are mainly metallic, and you won't be able to probably use most of the acids to digest your sample. So what we've been reverting to what we call them a soft extraction or a base type digestions. There's so many examples available on the PerkinElmer website through various applications For example, how to digest tissues, tomatoes, plants, and so on, how to digest plastics, or how to prepare plastic that have been embedded with nanoparticles, and you want to run for those nanoparticles. So the sample preparation is slightly different than just putting that materials, adding acid, and digesting it through the commonly used digestion methods.
Someone asks if you could please tell them what is the method detection limit for the water treatment plant measurements.
I would like to have more information about what he means by that.
Okay, well, maybe that's a question you can handle offline.
Yes.
Follow up with that person. Do you think it's possible to use this technique as a low-level detection of chloride using a simple silver nitrate test and measuring the silver chloride particles produced?
I don't see why not. It's a good example. I mean, it's a good thing. So in environmental samples, silver, after the dissolution, if we go, as I showed in the environmental section, silver tends to dissolve, and then it reprecipitate as silver chloride, and that reprecipitation of silver chloride basically shows up in the 20 nanometer range. So we basically can see that precipitation. So I don't see why not. I mean, it's really worth trying.
We have another silicon dioxide question. What is the recommended internal standard for silicon dioxide?
Well, I probably want to be able to answer that question in a slightly different way. Because we are doing single particle ICP-MS, and when we are doing that techniques, we basically focusing on one analyte to be able to understand. So matrix matching become a very critical component of the equations because we are not using a direct internal standards to it. So what we've been using is that I will take the sample, whatever that sample is, and I will add to it, for example, gold nanoparticles, and I would look at the average intensity of that pulse generated from that particle. And I will compare that to the standards. And we'll be able to compensate the matrix to be able to validate that the signal generated from that sample is equivalent to what we've been running on the standards. So it's slightly not a direct internal standard as we are doing a regular ICP-MS.
Someone asks whether it's possible to detect and size multi-element nanoparticles using ICP-MS.
It's a great question. What we've been doing, because well, I can say a few things. I can say we might be able to do so. At the current stage, what we've been doing, for example, in the gold and the silver example, or even before for the titanium and the lanthanum, we can slightly hop fast between the cerium and the lanthanum, and be able to look at the particle number. So we use the particle number as an indication to as if these elements are available in the same particles. Now we can do some fast scanning between two analytes to confirm that they are basically coming from the same particle. So the short answer, it might require a little bit of work, but yes, there is a work around it to be able to get that confirmation of these two analytes being present in the same particle.
Someone asks, what is the method to digest mesoporous silica nanoparticles? What acids should be used?
I mean, the instrument allows you to run the silica as is. You don't probably need it to. I mean, it's basically the mesosilica particles are particles, and you don't need to digest them. You just have to have them in the suspension, and you just analyze them directly. So there's no need for digestion. And I might want to add a little bit. I mean, I use always nanoparticles. The instrument can go to a micrometer size particles. Okay, so just keep that in mind. So we focus a little bit more on the nanoparticles because it was developed for nanomaterials. But there's nothing stopping us from using it for an application that have a micrometer particles. And when we're going to do the single cells, I mean the cells are in the micrometer range.
Someone asks whether they can perform single particle ICP-MS on an older DRC2.
It's a common question. We always do. So we always do receive that questions. The limitation of the DRC2 is again in the data processing and the data acquisition speed. The short answer: yes, you can. However, your data are broken, and your data will require manual interpretation for you to be able to achieve some sort of level of information. And if you are to try that, my advice for you is you will have to dilute your samples a lot compared to when we are using a microseconds dwell time, because that's not going to be able to allow you. You're going to be basically integrating just intensity of the pulse and not the area of that pulse or the amount of mass in that pulse. You're just going to say, okay, intensity. So to avoid running into what we call them coincidence or duplicates or clusters information, things like that, you will have to delete your sample for an extended a lot.
I think we're going to have to wrap it up. We've run out of time. Thank you again, Chadi, for your fascinating presentations and answering so many questions. And thank you, participants, for being a great audience. Be sure to check CNEN or CNEN Online for information on the next edition of CNEN webinars. Thank you, On24, for technology and production services, and thank you, PerkinElmer, for the sponsorship that made this interactive webcast possible. For CNEN webinars, I'm Ann Thayer. Goodbye. Hello, everyone. Welcome to today's webinar. I'm Ann Thayer, CNEN Senior Correspondent, and I'll be moderating today's event. This webinar is entitled Single Particle ICP-MS Overview of Environmental, Industrial, and Biological Applications, and it's being sponsored by PerkinElmer. CNEN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with CNEN's mission to provide news and analysis